So I'm pleased to speak to you today about two undertakings in drug discovery in which I am involved, in both cases directing the science of small companies, one of them called Interex, the other called Searvir. Interex is an undertaking in computational chemistry, where Interex is located in Menlo Park, Berkeley, with offices also in Pittsburgh, Moscow, and Tel Aviv. The challenge of computational chemistry, illustrated in this slide, is of discovering a ball and socket or hand in glove fit of a small molecule to what is usually a protein target, and in many cases an enzyme, and often the small molecule will be an inhibitor of the enzyme. Now, in current practice, and in the best case, in pharmaceutical companies around the world, this process of drug design begins with a co-crystal structure of a lead compound with the protein target, on the basis of which an improved structure of the lead compound is designed, synthesized, tested, and if successful, then the process is repeated in a very large number of times. It is an expensive process because of how many times it needs to be done, and time consuming as well. Typically, hundreds of chemists will make thousands of molecules to arrive sometimes, but not always, in fact, not even often, at the desired drug product. It has been hoped for a very long time that this arduous process and not often successful process might be augmented or even replaced by computation. The idea is that one might compute and not have to perform chemistry as often and arrive at better molecules. The computation requires knowledge of the forces between the small molecule and the protein target. The force fields involved are, in every case except what I will shortly describe, classical, which is to say electrons are represented as point charges. The problem with the current force fields is that they don't work. The force fields are simple and rapid, but they do not give a successful result. And here at the bottom right, you see a plot of the experimentally determined energy of interaction of a small molecule with a protein target. And on the X or axis or abscissa, the computed energy of interaction. And it's obvious from the scatter of points in the diagram, the results are essentially random. I will describe now a solution to that problem developed over the past 20 years through the efforts currently underway at Interex, whereby electrons are represented not as point charges, but rather as they occur in nature, as clouds. The representation then is a quantum mechanical in nature, and the force field we refer to as a quantum mechanical polarizable force field. I'll explain why polarizable in a moment. The important point is that a polarizable field can represent interactions that require polarization, such as van der Waals interactions, which are responsible for the contacts between hydrophobic surfaces of small molecules and proteins. And as I will show you, this is an approach which successfully computes interactions in water of ligands with proteins in a manner which is not only better, but really the only way that it can presently be done in a reliable manner. The quantum mechanical energy includes two classical terms, the interactions of the bonded atoms, and also the electrostatics, the charge-charge interactions, but then in addition, the three quantum mechanical terms shown here, electron exchange, electron dispersion, and induction. The idea of polarization is illustrated at the lower right for a water molecule. And what you see in green and yellow are the electron clouds, green where they're concentrated, yellow less so, surrounding the two hydrogen atoms and the oxygen, the two nuclei of hydrogen atoms and the nucleus of an oxygen atom in a water molecule. What you see here are how those electron clouds move about during the course of an accurate quantum mechanical simulation of the molecular structure. The quantum mechanical polarizable force field is more complex than the current classical force field, but most importantly, it gives precise results as indicated by the vertical axis and the quantum mechanical polarizable force field, a rapid result as shown on the horizontal axis. A full quantum mechanical calculation would require on the order of 1 trillion years or more with current computing capability. The quantum mechanical polarizable force field, QMPFF, represents an approximation which was discovered over this lengthy period of time that I've mentioned at InterX and its predecessor, which can be done in an acceptable length of time, currently about 8 hours for a computation, almost as fast as a calculation with a conventional force field, which gives, as I have mentioned, a much less accurate, indeed inadequate result. Does the QMPFF work? Here I show you one example. What you see in the gold balls or circles are the results of our calculations, where we have plotted the computed energy of interaction on the vertical axis, the experimental shown on the horizontal axis. You see that the gold or orange balls lie very close to a straight line, showing a very accurate result. In contrast, the pink triangles and the purple squares show the results of calculations with the best available classical force fields, clearly inadequate for the purpose. This is a kind of retrospective study. The next slide shows one example of a prospective study where we tested our ability to actually do drug development, work that has been completed just in the last few weeks. We took as an example inhibitors of poly-ADP ribose polymerase. Such inhibitors are already approved for monotherapy of BRCA mutations and in use in over 100 trials in combination with other therapies. In the course of only 8 months, we could test with the use of QMPFF greater than 60,000 molecular designs. We synthesized only 26 compounds during the course of those 8 months and arrived at a molecule which has 100-fold higher affinity and efficacy than any of the existing inhibitors presently in clinical trials. With that, I'll turn to the example of I'll turn from small molecule drug development to the example of nucleic acid drug development, and in particular, silent interfering RNA. And at Servier, a company located in Israel, we have applied this approach to human respiratory viral disease for the purpose of both prophylaxis and treatment of disease. siRNA molecules present a particular challenge. The small molecules we've spoken of until now, small molecules are typically a few hundred Daltons. siRNA molecules, in contrast, are many thousands, 14, 15,000 Daltons, and bear as many as 50 negative charges, which would ordinarily prevent their penetration, their entry in cells, their crossing the lipid bilayer membrane of any cell of any type. The solution to the problem is to exploit membrane dipole potential. This is an electrical potential difference between the surface of the membrane at the membrane-water interface and the center of the lipid bilayer. Because of the low dielectric constant of the lipid core of the center of the bilayer, there results an extraordinarily high electric field on the order of 1 billion volts per meter, never exploited in the past for medical applications until what I will tell you now. In order to do that, we have employed a derivative with what we call a molecular nanomotor, or M&M, a molecule which is based upon cholesterol but with halide atoms attached to create a very large electrical dipole moment. Here you see those M&M's molecular nanomotors attached to a double-stranded, so a double helical siRNA molecule. The blue and brown represent the siRNA double helix. What you see on the left here are the results of an experiment in which an siRNA without those molecular nanomotors attached, applied to melanoma cells in culture, and then after washing, all of the fluorescently labeled siRNA is gone. On the other hand, when the fluorescently labeled siRNA was conjugated with the molecular nanomotors in the manner shown in the previous slide, then it is all taken up in the cells. And the uptake is rapid. Almost all of the siRNA taken up within an hour. The reason why it's all taken up and retained is because the molecular nanomotors are attached through disulfide bonds to the siRNA, and they are cleaved in the reducing environment of the interior of the cell and trapped in the cytoplasm. Here you see that when the siRNA contains a sequence complementary to the messenger RNA for epidermal growth factor, and then the expression of EGFP is drastically reduced, as shown in the two green bars on the right-hand side of the slide. On the other hand, with an irrelevant sequence or none, then EGFP expression is unaffected. Finally, in the last two slides, I'll show you the application of this approach to SARS-CoV-2, experiments that have been done in only the last couple of months. So this slide shows a study done in vitro, so introducing the siRNA in the manner I described into cells in culture infected with SARS-CoV-2, and the result with either an siRNA complementary in sequence to the RNA-dependent RNA polymerase gene of SARS-CoV-2 or complementary in sequence to the leader RNA of SARS-CoV-2, there is a 3-log reduction in viral load within 24 hours. The last slide shows an application to a non-human primate, to the African green monkey, whose respiratory system is most similar to that of humans. And if you look at the panel on the left, for example, you see the results of an oropharyngeal swab. You see that there's a 98% reduction in viral load 2 days after infection, and still in effect 6 days after infection, with the particular dose of the MNM derivatized SARS-CoV-2 complementary siRNA that was introduced in this case. It was actually a rather low dose, and an even greater effect and longer lasting one will be obtained with more drug. These experiments show that the drug is non-toxic, perfectly safe in the monkey, and as I have described, highly effective. We look forward to development of this, we hope, in a timely manner for addressing the current pandemic. And with that, I thank you for your attention.